So if you look at the antibody structure that you see here, you can see the heavy chains and light chains. You can see the disulfide bonds, the 2 disulfide bonds connecting the heavy chain, the 1 disulfide bond each connecting a heavy chain to a light chain. You can see our constant region, which we're going to end up talking quite a bit more about today. You can see that the antibody is sort of divided up into these rectangles. And those rectangles that we discussed last time are, in fact, each immunoglobulin domains. Those immunoglobulin domains contain beta sheets that are held together by a disulfide bond, and there are loops at the end of those beta sheets. If you look at the view on the top, you can see a few different beta sheets, and you can also see the loops that are connecting the beta sheets sitting up at the top of the immunoglobulin domain. I'm going to be pointing out those loops again in a later slide, so I wanted to refresh your memory about them. Yep, Mark.
So on the previous slide, it showed like the one region where like the bend happens, like without any vapor.
Yes.
Is that just like a connector?
It's called the hinge region, and it is not officially an immunoglobulin domain. Yeah, it's really more for a connection. So as I mentioned, we're going to spend a little bit more time talking about the constant region later today, but there are a couple of parts of this section of the antibody that binds antigen that I didn't quite get to last time. I mentioned that the sections of antibody that are made up by the light chain and a portion of the heavy chain are sometimes known as the FABs, or the fragment for antigen binding. These are the parts of the antibody that bind to antigen. They're identical on either end, and this is where we're actually going to make contact with the antigen based on sort of the three-dimensional shape that is made by that heavy chain and light chain together. That binding does require both the heavy chain and the light chain in order to be specific for the antigen. And you can see this at the top, where you can see that the antigen is sort of shown as binding between the heavy chain and the light chain. You can also see an antigen shown here making contacts with both the heavy chain and the light chain across a surface on the bottom. Specifically, the parts of the heavy chain and the light chain that are making those contacts are those loops at the end of the immunoglobulin domain. There are 3 loops on the end, and so this one, and this one, the blue ones, they have a little L subscript. They're from the light chain because of the L. And the pink ones are from the heavy chain, and so you can see that those 3 loops from both the heavy chain and the light chain actually are the things that are coming in contact and binding to that antigen. We called this bottom portion of the antibody the constant region. And we talked about how there were 5 different kinds of constant regions and things like that. The portions of the heavy chain and light chain that are binding to the antigen will have to be different between antibodies that bind different antigens. So the ones that bind to one particular epitope of HIV will have to be unique. The ones that bind to a different epitope on Ebola will have to be unique. And so oftentimes we refer to these portions of the antibody as the variable regions, because they are much more variable and they vary quite a bit from antibody to antibody. And in fact, you can see this actually says VL for those, and VH. It's because they're the heavy chain variable and heavy chain light, or light chain variable, that are binding. If you were to take the proteins, we take a bunch of antibody proteins, and you were to do sequences of their amino acids, you could make them, you could put them in a line. You could say, at position number 1, what amino acid does most antibodies have? At position number 2, what does most antibodies have? And you could go through all the amino acids. At some parts, like in the constant region, you'd pretty much always come up with the same amino acid. In other parts, you'd come up with more variation. And in fact, most of the variation would be, unsurprisingly, in the variable region. If you actually made a graph of this, these two graphs, this is actually showing the heavy chain on the left, this is showing the light chain on the right, and these are just showing the variable regions. And so what you will notice is that among the variable regions, the variation is not constant. There are some areas that don't vary too much. And there are some regions that vary a whole lot. You will see that there are 3 of those regions in the heavy chain. There are 3 of them in the light chain. We often refer to those 3 regions as the hypervariable regions. And so there are 3 hypervariable regions in the heavy chain and 3 hypervariable regions in the light chain. And so sometimes we will call them HVR 1, 2, and 3 for hypervariable region. It turns out those 3 really, really variable regions are the loops at the end of the immunoglobulin domain. They are the things that are making contact with the antigen. They determine which antigen is complementary to the antibody. And so sometimes we also refer to them as the complementarity determining regions, or CDRs. And so sometimes we'll see them listed as CDR1, CDR2, and CDR3 for complementarity determining region. They're the 3 little loops, 3 little fingers, that are actually going out and touching the antigen and indicating which antigen that antibody is complementary for. So this is our antibody molecule. If you go back to the survey that you did before the first day of class and look at some of the information that you wrote about antibodies or that your non-science friends wrote about antibodies, and if you, in fact, generally read sort of about the immune system and about antibodies from non-scientists, antibodies are superheroes. The things they can do are amazing. And things become a little more complicated when you realize that the antibody is simply 4 protein chains held together. It's just a protein. Sometimes people will, it's going to destroy all the microbes. It eats the microbes. It's 4 protein chains. It can't eat anything. You know, like the types of activities people want to give to this antibody become a little bit of a challenge. And so a big part of what we're going to talk about today is how it is that this set of 4 protein chains, this molecule, can actually do something about a microbe. Yep. We're going to learn, a big part of what we're going to talk about today are the effector functions of antibodies. What antibodies do. When I was starting on the job market, I had to give a lot of talks, to get a job, and one of the things I learned was that effector function is a word that only immunologists use, and because people are like, you have to get rid of the jargon, and I'm like, that's not jargon, what are you talking about? Like, I forgot that was even a thing that only immunologists say. So effector function is something we're going to hear a lot this semester. Effector functions are just the ways microbes get dealt with, or the ways, like, the cell or the molecule does something against a microbe. So officially, we are talking here about the antibody effector functions. There are listed here 6 antibody effector functions. Sometimes I usually will talk about them as the 5 because there are a couple that are really similar. So whether we want to call them 5 or 6, that's fine. So we're going to need to talk through what these different effector functions are. Happily, you already know 2 of them. So, have we seen antibodies anywhere in the course thus far? Yes, Mark. Complement. Remember when we learned about the classical pathway of complement? The first step of the classical pathway of complement was antibody binding. So in fact, one of the effector functions of complement is that it can, or of antibodies, is that they can start complement. So effector function number 1 of antibodies is they can fix complement. So antibodies can bind to antigen on some sort of surface and start off the classical pathway of complement. So that one's pretty easy. You already know that one. The other one you already know, but don't realize you already know. When we talked about complement, I also introduced you to a vocabulary word called opsonization. So opsonization was this process where complement fragments coated something and made that thing more likely to be phagocytosed. I told you it was like butter. It's coating something to make it more likely to get eaten and to look more tasty. The second big function of antibodies is that antibodies also can do opsonization. So if you coat something with antibodies, that also makes it more likely to get phagocytosed. And so here we have some microbe that's coated with antibodies, and that will enhance the likelihood of that microbe getting phagocytosed, just like coating with complement would do. One thing that this implies is that there are cells, specifically phagocytic cells, that have receptors that can bind to antibodies. When those receptors bind to antibodies, they are binding to the Fc portion of the antibody. As a result, those receptors are called Fc receptors. There are tons of types of Fc receptors. You can see that there are things like Fc alpha receptor. It binds to IgA, the one with alpha. There's our Fc gamma receptors. They bind IgG, the one with a gamma. You can see there is an Fc epsilon receptor, binds to IgE. So all of this does sort of tie together. What I want you to notice here is that there are a lot of different types of Fc receptors, so there are a few different functions that can happen with Fc receptors. Opsonization is one of those functions, but it's not the only one. So those are the two. We already got 2 out of 5. You already knew them. Easy. The third one that I want to mention to you is one that people who work on viruses think about all the time. So usually when you think of virology, the next one I tell you about is the most important. And in some ways it's a pretty easy one to understand. So for me it's the one I like because it's the one I think about the most often and it just makes a lot of sense. And this is a function of antibodies called neutralization. So antibodies are able to perform this function of neutralization. On the left, you can see cells from a person that are getting infected with a virus. That virus is binding to a receptor on the host cell, which lets the virus into the cell, lets the virus do its stuff. If, however, there are antibodies present that are able to perform neutralization, those antibodies can bind to the microbe and physically block that microbe from interacting with its receptors and getting into the cell. And so neutralization is basically binding to the microbe. It's not binding to the cell. It's binding to the microbe and physically blocking further interactions. In some ways, neutralization is a fancy word for steric hindrance. It's just that the antibody is binding to the virus and getting in the way of other things happening. You can see an example here. So this is actually a structure of a rhinovirus. It's one of the causes of the common cold. And you can see that this blue protein has to interact with a receptor in order for that virus to get into the cell. If antibodies bind to that virus, the antibodies are shown in green. They can actually get in the way. It could be because they bind to the same site, or it could be because they bind nearby and are just blocking the receptor interaction because the virus can't get close enough to bind to that site. And so this is sort of this idea of neutralization. Oftentimes when people are doing virology studies and are measuring immune responses to a virus, instead of trying to measure all antibodies to the virus, they do special types of experiments that let them measure how many neutralizing antibodies instead of all antibodies in total. Yep, Nick. No, bacterial cells absolutely can be neutralized by antibodies. Bacterial cells often can be active outside of cells. They can replicate outside in the extracellular space, and so neutralization is going to be less useful because the really important part here is that you're stopping this virus from getting into a cell. Viruses are obligate intracellular parasites. They have to get into a cell in order to replicate, and so neutralization tends to be really important because they stop that important step for viruses. But there are absolutely ways that bacteria can be neutralized too. That's why we think about it as being so important for viruses. Did you have a question, Vanessa?
So there's specific different kinds of antibodies that can do these different things, so you have to neutralize
Yep.
them. And they all have different variable regions as well?
That's the second half today.
Okay. So it just seems like a really insanely vast amount of
That's later. All that's coming. So Nick asked a question about bacterial neutralization and whether that was a thing. And it can be. It's a little bit less important than we think about viruses. However, some bacteria cause disease because they make toxins that hurt our cells. And very often, we can see toxins be neutralized. So often, a toxin may bind to a receptor on a cell that may let the toxin into the cell where it can do its damage. We can imagine antibodies neutralizing that toxin and blocking the toxin from interacting with the cell, and thus actually blocking the ability to cause damage to the cell. In reality, this is actually the thing that's unique about the 3 microbes that we saw in the von Behring and Kitasato experiment on Friday. I told you that they were really lucky with the microbes they picked in terms of how they got their experiment to work out. It's because all 3 of them caused damage by toxin, and in all 3 cases they were actually getting toxin neutralization with their antibodies. So what you will see as we talk about a bunch of different big Nobel Prizes is almost every one of them, I can tell you and they did it this way, and it worked, and they won the Nobel Prize. But if they had made this one small change in the experiment, everything would have fallen apart. So there's always a good role of luck in all of these stories. Yep.
So when we think of toxin neutralization, is it more like thinking of the antibody binding to a small molecule that can cause damage to the cell?
Yeah, exactly. Okay, so we got 3 functions now. They're not too terrible. The 4th function is the function that nobody ever likes because they say it's lame. So the 4th function you're going to look at and you're going to be like, what does this do? Like, who cares about this? But it is actually pretty important. I promise it's going to not look important at all, but it is. And the 4th function of antibodies is the ability to form antigen-antibody complexes. And so if you have antigens and antibodies present at the right concentration, because each antibody has 2 places where it can bind antigen, and because many antigens have multiple epitopes, you can sometimes get these great big complexes of many antibodies and many antigens coming together and binding. And that can form really big clusters of proteins, really big clumps of proteins. That can be useful or that can be important because sometimes what will happen is that this will actually bring that antigen out of solution. It will basically precipitate the antigen and pull it out of solution. And that could be useful because it won't diffuse a whole lot. And so these are known as antigen-antibody complexes. And the ability of antigen-antibody complexes to be formed is particularly important. Oftentimes, antigen-antibody complexes will get cleared by phagocytic cells. So eventually, we will get rid of the antigen-antibody complexes. So this often does tie into some other stuff. I will also point out that antigen-antibody complexes are really important parts of a number of different diseases where they can actually start to clog up parts of the body. And so there are situations where in kidney disease, you have antigen-antibody complexes actually getting in the glomeruli and clogging up the glomeruli. There are some similar diseases where you see them in the alveoli. There are similar diseases where you see them in the joint. And so antigen-antibody complexes are also really critical to know about because they do have this big role in disease pathogenesis. Yep.
So in the previous slide, what did it mean in that picture when it said like naive versus
We'll talk about that later. Okay, so we're up to 4. We got 4 functions of antibodies now. Yeah, Vanessa. It's really about making the antigen a bit easier to clean up. So the next function of antibodies has sort of a special name, and there are a couple different ways that you can think about it. So this function of antibodies is called Antibody-dependent cellular cytotoxicity, or ADCC. Later on in the semester, I am going to mention ADCC. And I'm going to say, you guys know about that, you remember that, you've heard of that, right? And you're all going to be like, who? You did hear about it, right here. ADCC is when antibodies activate a type of cell called a natural killer cell. And so what is happening in ADCC is that we are seeing activation of NK cells. I'm going to give you a very sort of simplified picture of what's going on here, and that's because we need to talk way more about NK cells for a less simplified version of this. We can imagine that there is an antigen that is on the surface of a cell. My favorite example of this is if we have a virus that is being produced by the cell. Sometimes the virus has its proteins in the cell's membrane. There can be antibodies that can bind to that antigen. NK cells have Fc receptors that can recognize the Fc portion of those antibodies, and then the NK cell will kill this cell that is attached to those antibodies. And you can see that here as well. The antibody can bind to some type of cell target. The NK cell's Fc receptor will interact with the Fc portion of the antibody, and the NK cell will kill that cell. And so you can see this is antibody dependent, because you have to have an antibody to make it work. There's a cell, an NK cell, and it kills another cell. So that's where we get antibody-dependent cellular cytotoxicity, or ADCC, from. So this is, again, sort of the 5th function of antibodies. Yeah, Nick?
Is this in relation to only your own cells?
No. The other function that is shown on the next slide is somewhat related to this one. And that's why I always learned these as 5, but your book says they're 6. And like, yes, technically this is different. So sure, I'll give them 6. I'll take 6. Fine. We can also see antibodies binding to other Fc receptors. In this case, these Fc receptors are not on NK cells. In this case, the Fc receptors are on a different cell type called a mast cell. And if those antibodies bind both that Fc receptor on a mast cell and an antigen, like the antigen that may be found in this parasitic worm, that will activate the mast cell to secrete its products that are in its granules. And so the activation of mast cells, where the mast cell takes all of its granules and loses them by expelling their contents, is the final function of antibodies. So we've got these nice antibody functions, but Vanessa started to ask a question earlier about sort of variation between which antibody does what and sort of what's going on with all of that. And that's the other place that I want to take this today. We talked last time about antibody isotypes. I told you that there were 5 different isotypes. You will see today that that was a little bit of a simplification. They can be 5. 5 is fine. And these 5 vary based on their constant region. And so I told you there were 5 different types of constant regions, M, D, G, E, and A. They are encoded either by the mu, delta, gamma, epsilon, or alpha genes. And so what we need, and in fact, they differ in this Fc portion. It turns out that different isotypes of antibodies are good at different functions. And so there is a relationship between these isotypes and the functions that I have just told you about. I'm going to make a slight diversion for a couple of slides, and then after that, we are largely going to spend the rest of the time today talking about the unique properties of each of these 5 isotypes, which will tie into the functions that I have just told you about. So the next slide is going to cause some problems. So I'm going to give you the preface to the next slide first. I need to tell you the information on this slide. What I want you to know when we leave this slide is that this exists. This is a thing. Those of you who have taken a fair amount of molecular biology or genetics are going to look at it and be troubled. And you are going to look at it and say, no, 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 no. No. You're going to have 1,000 questions. Guess what? All those questions get answered later in the semester. Right now, we are not going there. However, you need to know that this is a thing. Okay? So I understand how many of you are going to freak out about this and how this is going to seem to break all the rules. Yep, it does. We'll deal with that later. So it's okay. It's okay. So antibodies can change over time. There are 2 specific types of changes that are observed in an antibody response over time. So if you were making an antibody response to a particular antigen, and if I went to your body every day and I took some antibodies and I measured the antibodies you had every day, there are 2 changes I would see happening over time to your antibodies. To be honest with your question mark earlier about naive versus memory, it's kind of because of this too, but again, not going there. One of the changes that we can see is actually that the antibody will switch the isotype. So all antibodies start out as IgMs, or all antibody responses start out as IgMs, but we will in fact see switches to other isotypes. So in fact, we can see responses where perhaps a particular pathogen would be combated really well by IgE. We can see a situation where the same variable region is put with a different constant region, thus allowing you to have an IgE response to that antigen. I see all the faces already. I see it. The other thing that we can see is that there are changes in the binding strength of the antibody to the antigen. The antibody is actually able to change and have tighter binding as the response goes on. This is referred to as affinity maturation. The affinity for antigen matures and gets better. And so we can also improve our variable region over time. All of this comes up later in the semester and gets like its whole own day. Yes?
So when you say variable regions over time, like literally the amino acids that make up the change?
Yes.
Okay.
Like I said, I see all the faces. It preempts another set of questions. If I don't tell you about this, then suddenly you have this other set of questions. So I have to tell you, but I also know it freaks everybody out. I also want, just so you know, differences in antibody isotypes are not the only kind of differences that you can describe in antibodies. We are going to talk about isotype differences today. And here you can see some isotype differences where we have an IgE versus an IgG. They have different constant regions. We can talk about antibodies that are the same constant region, the same isotype, but that have different variable regions. Those are known as different idiotype antibodies. You can imagine it as being something complicated like I was just talking about, because you're all making faces still. Or you can imagine two antibodies with idiotype differences would be an IgG that binds to HIV and an IgG that binds to Ebola. Like, they just bind to different antigens, but they're both IgG. That's actually all an idiotype difference is. Different variable regions, but both the same isotype. We can also see individual differences from person to person that may be due to polymorphisms in different people, just general genetic changes from person to person. So here you can see we have Sherry's IgG against antigen B and John's IgG against antigen B. Those are known as allotype differences. And so we can also see differences in allotype. Today we are largely going to be talking for the rest of our time about isotype differences. But you can use other terms to describe some of these types of differences. Okay, so here are the isotypes. So this is a table from your textbook. We're going to be coming back and forth to this table a bunch of times. Across the top of the table, you see different isotypes listed. And you will see that in reality, there are like subtypes of G and there are subtypes of A. We don't really care right now. Don't get too stressed about that. But yes, in reality, that is the truth. And then you can see a whole bunch of different types of variables or things that we could think about as being different between the isotypes. And so we're going to be focusing on the unique aspects of some of these isotypes, what makes them special. When I told you about the isotypes, I told you that there is this mnemonic that people use to remember them. MDs give everyone apples. I always think about them in that order. It's weird to me that I'm going to present them out of that order, but this order makes more sense. So we're going to do it this way. So the first one we're going to talk about is IgM. If you look at this table where I have put a red box around information about IgM, where does IgM seem to stand out? There's one particular place where IgM really stands out. Yeah, Paula? By its molecular weight. If you look at the top, which is molecular weight, you can see IgM's molecular weight is way bigger than all the other ones. And so there's already something interesting going on here, making IgM very unique. The reason, so you can also see, if we look at, say, serum level, it's sort of in the middle. If you look at half-life, it's sort of in the middle. The one place where it's really special is molecular weight. The reason why IgM is special is because IgM is generally found as a pentamer. You will find 5 copies of an identical IgM molecule bound together by a J chain. The J stands for joining. And so this results in IgM that we find in serum having a much higher molecular weight because it really contains 5 antibodies, whereas the other ones contain 1. This is awesome because IgM now has 10 sites where it can bind antigen instead of only having the 2. And so it can bind to antigen pretty well, you can imagine, because it is found as a pentamer. You can also imagine that it can make some weird shapes because of this. When IgM is a pentamer, it can bind to multiple copies of an antigen on the surface and make this really cool structure on the surface of a microbe. And that structure is the best possible structure for complement to bind, for C1 to bind. And so IgM stands out among all of the other isotypes in that it is the best at fixing complement. It is by far the best at starting the complement cascade. IgM is also made first in the response. It's made very early in responses. And so sometimes when we're thinking about trying to measure how far along an immune response is, knowing about IgM amounts will be important. And that'll be important when we talk about immune response kinetics later. So the second isotype we're going to talk about, oh yeah, Mark?
Is the J chain like a separate thing?
It's a separate protein, yes. Yes.
Are we going to talk about that now?
Not particularly. Okay. So the second isotype that I want to talk about is IgE. So if you look at the data on IgE, what do you see as an important, or sort of standing out about IgE? Yep, Ashley. The serum level is really low. If you look at the normal serum level, it's super, super low. The reason why the normal serum level is really, really low for IgE is because it's found somewhere else. It's not usually in the serum. Instead, IgE spends most of its time bound to mast cells. So in fact, mast cells have these Fc receptors that bind specifically to the IgE constant region. They are the Fc epsilon receptors. And mast cells just get IgE covering them. And so we don't have free IgE floating around. All of your IgE right now is on mast cells, already bound to those mast cells. That means if the antigen that binds to that IgE ever comes around, it automatically can bind, and that mast cell can automatically make a response. That's why when you have an allergic response to something, it happens so fast, is because you have the pre-bound IgE bound to the Fc receptor, and you only need to have the allergen, and suddenly the mast cell is ready to go. So that's IgE. And so IgE is really the best at activating mast cells, and to be honest, it is the only one that activates mast cells. None of the others that can activate mast cells. And so whenever you're having an allergic reaction and you're angry it's IgE's fault. You can blame it all on IgE. So the next antibody isotype I want to tell you about is IgD. This is the figure about IgD in your textbook. However, instead of telling you about this figure, I want to actually tell you about this figure about IgD. This is IgD, the version of IgD from the last version of your textbook. So this is the past version of your textbook, the old one. And if you look at IgD, and you look at anything that stands out about IgD, what do you notice on this version of the figure about IgD, if anything?
I noticed that there's a difference from the last one.
Okay, yeah, we're going to get to the difference from the last one. But what do you notice about this one? IgD's fun. Yeah, Vanessa? It has all negatives down here. For this list of what function it does, like before we were seeing mast cells, oh yeah, IgE. Complement, oh yeah, IgM. Here, if you look, it's all negatives. And so for most of my life as an immunologist, the function of IgD was no known function. It was maybe an evolutionary relic. We can use it to track status of immune response really nicely. We know nothing about what it does. And most of my career as an immunologist, that's what we know for IgD. If you look at my old exams and look at the keys, when it asks about like functions of different isotypes, you will see lots of answers of IgD, no known function. One thing that we have known for a while is that in some patients with some autoimmune conditions, you tend to find more IgD than normal. I got nothing for you. In the past few years, there has been a function for IgD that has been described. This figure from your book doesn't make it sort of as clear. What we now think that IgD is able to do is induce basophil degranulation. So it does the same thing that I just showed you about IgE and mast cells, but it does it with basophils. The problem is the basophils are very poorly studied and rarely studied. We sort of joked when we did the blood lab that your patient only had one of them, so it was good that you couldn't find one so they didn't lose it. And so it's incredibly hard to study basophils. It's incredibly hard to get them out of a mouse to actually look at what's going on. And so maybe the reason why we never knew a function for IgD is because we didn't have the right tools to study it. And so this has been something that people are now starting to realize, is that IgD may be really important for basophils, while IgE is really important for mast cells, but this is much newer information. Yep?
What about the presence on membrane of B-cell naive?
Yes, so when we get to B-cell development, IgD will be an important marker of a certain stage of development. All right, so I've done M, I've done D, then I went to E. And now I'm going to do A, because I'm just really all over the place with my letters. And so here you can see IgA. So if you look both at the sort of data on molecular weights and serum levels, as well as in sort of looking at the functions, what stands out to you about IgA? Yep, Tony. So there's a range of molecular weights. That's one thing that should stand out. Yeah, Mark. Mucosal transport is the other thing. IgA can be found in two different forms. One of them is just normal old IgA, like this. IgA can also, however, be found in a form as a dimer, where we have two different IgA molecules bound together. They are bound together by a different J chain, their own J chain, that allows us to make this dimer. And so this is sometimes known as Dimeric IgA or Secretory IgA. The really unique thing about IgA is IgA's location. So IgA, like any other antibody, is secreted by some B cell into the blood. However, that antibody then binds to a special Fc receptor, is brought into a vesicle in a cell in the epithelial layer, and is then delivered across the other side into mucosal areas. So if you think about the inside of your GI tract, there's no B cells there. But in fact, there's a whole bunch of IgA that got transferred across the wall of your GI tract. Similarly, your lungs are full of IgA that got transferred across the epithelial wall. And all of your other mucosal surfaces all are full of IgA. This process is called transcytosis. We're going across a cell. You know, we don't want this to get degraded or anything bad to happen to it, and we're just doing FedEx to move it to a different location. That happens because of this Fc receptor that in fact binds to the special J chain that IgA has. It's J chain binding. And so IgA is largely going to be functioning in this case, we're seeing the pharynx sort of in the actual windpipe, where the IgA has actually gone across, away from the B cells, just to hang out in that location. And it can be really useful for neutralizing pathogens in mucosal sites before those pathogens even sort of breach any barrier. And so in some ways, IgA is almost like part of the barrier immune response because it's in some of those tissues that are in contact with the outside world. Yep.
So does that mean like only or mostly dimeric IgA is found in like mucosal?
For the most part, yes. So if we look at IgA, like I said, the thing that's unique about IgA is the anatomic locations where it is found. And so you can see here an image of a person and different isotypes in terms of where they are. What I want you to notice is that IgA is largely found in mucosal tissues and only in those mucosal tissues, and it's sort of the only one. Those tissues are just yellow and aren't any other color too. And that's really where the IgA is. So you can see, you know, nose, mouth, you can see eyes. When you cry IgA. Lungs, GI tract, all that good stuff. IgA is also found in breast milk. It is the only isotype found in breast milk. And so when babies are breastfed, they actually get IgA from their mom. So the IgA is transported across the epithelial cells into the parts that are involved in lactation, the whatever gland, mammary gland, but I feel like there's another word, and the only word I can think of is actually, I know is the word for tear duct, and so like, I just know whatever word is in my head is wrong. Babies also get a lot of IgA from their moms if those babies are breastfed. Yes?
Have there been any studies done correlating immune responses in people who were breastfed and who were not? I'm sure there are, but are they conclusive?
There are many of those studies. They are not particularly conclusive because there are a number of other variables that happen within those types of studies. However, one other fact that we should talk about with IgA is that the most common immunodeficiency is selective IgA deficiency. So here you can see a bunch of different types of populations and the number of people who have selective IgA deficiency per million individuals. And so if you think about any type of immunodeficiency, loss of some part of the immune system that you might see, this one is by far the most common. And you can see there is a big amount of variation between different groups. Details are sort of not super clear on that. It seems as though people who are IgA deficient are healthy. We have seen no defect in these individuals. It's possible that that's because of modern hygiene and that maybe in the past it was a bigger problem. There are some epidemiological studies that maybe associate this with lung disease or celiac disease, which you can sort of imagine since those are both mucosal tissues that would be impacted. And some people have wondered, well, why has this not been selected against? Why have we not gotten completely rid of selective IgA deficiency? And if you think about it, most babies have IgA because they're of breastfeeding. So if they have IgA deficiency, it doesn't matter. They got mom's IgA. And so it's only later in life when they've gotten sort of past that extreme susceptibility to infection that they're actually really lacking IgA. And so that may be why this has sort of persisted in the population over time. There's a lot that we don't understand about this, but it is numerically sort of one of the most common immunodeficiencies, one that people may have seen or heard of. So we got 4 isotypes down. We got to talk about number 5. Our 5th isotype is IgG. You can see some information about IgG here. So what types of things do you notice about IgG? Yep. It's the only one that crosses placenta. We'll come to that in a second. Yep. It has a very specific molecular weight, so we probably don't have any monomer, any dimers or trimers or any funny things like that. Yep, Lexi. Mm-hmm. Pretty good half-life, pretty long half-life. Yep. It's also found at the highest level in the serum. And so generally when people measure antibodies or when people do work with antibodies, they're doing IgG work because IgG is by far the most common antibody. It's really the one that's in the blood doing a lot of actions. And if you actually look at our functions, you can see opsonization, pretty good, okay with complement. It doesn't do the special mast cell one. But other than that, it kind of does everything. So no mast cells, but otherwise IgG does all the functions. And it's present in high levels. There are 4 different subtypes of IgG. They largely vary based on that hinge connecting domain and how long it is. This changes things like how easily the molecule is cleaved by proteases, which is actually what changes the half-life. But by and large, IgG is sort of the antibody. When people think about antibodies, they're usually thinking about IgG. Yep?
So the hinge domain is variable. Why
Yes.
is the size generally the same?
Because they were rounding on that figure. And there's also issues of amounts of glycosylation that's going on, so that also can sort of vary that number a little bit. Typically, when I think about IgG, one of the really big things I think about is that IgG is going to be indicative of a secondary response. So early in responses, we tend to make IgM. Late in responses, or when we're making a memory or secondary response, we tend to make IgG. And so if I wanted to measure whether you were making a memory response to some antigen, I would measure IgG. To be fully truthful, I could measure A or E as well, but they're present at such low levels, that would be hard. So I'm just going to measure the easy one that's present at really high levels, which is G. And so IgG is usually the thing that we do a lot of measurements of to measure progress of immune responses. IgG is also unique because it crosses the placenta, and it is the only isotype that crosses the placenta. And so in our unborn fetus, that fetus only has IgG coming from its mom. It will acquire some IgA if mom breastfeeds, but IgG is the only one that's going across the placenta. Again, this is happening through a process of transcytosis, where there is an Fc receptor that is actually carrying the antibody across the placenta in order to make sure that that antibody is present there. And so what you can notice is that in infants, by and large, right after birth, most of their antibodies are actually antibodies that have come from their mom through the placenta. It's IgG. Most of those antibodies are passively transferred antibodies. You can see that they'll also start getting some IgA. This will vary a bit based on whether or not they're breastfed. And they won't start making their own IgM or IgG until a little bit later. This is important for a couple of public health issues. So people sometimes get really angry about the vaccine schedule. Oh my gosh, why does my baby have to get a whole bunch of shots all at once? That vaccine schedule is actually set up so that the baby is getting their shots as soon as mom's IgG is gone, because if mom's IgG was still there, it would actually inhibit the vaccine. It would clear the vaccine. It would destroy the vaccine before the vaccine could actually start to do something. And right when the baby's immune system is starting to do something. So you don't want to give it too early. Mom's IgG is going to destroy things. But you want to protect that baby from harm as quickly as possible. And so this sort of information is a big reason why the vaccine schedule is set up the way it is. There are some types of infectious disease that are particularly problematic to infants. The most famous is pertussis, or whooping cough, where babies that are, very young, weeks old, often will die of pertussis. It's a really, really bad disease in little babies. And you can see that there's this problem because those little babies aren't making their own immune response yet. They can't be vaccinated against pertussis right away, and yet they are the most susceptible to disease. As a result, the CDC has actually recently changed the way they do some parts of pertussis vaccines. So everybody gets pertussis vaccines. However, pregnant women get an extra dose of a pertussis vaccine. And the idea is that those pregnant women will start making antibodies against pertussis and will pass them through the placenta to the baby so that the baby is covered right away. And so now pregnant women are given a pertussis vaccine during their pregnancy in order to take advantage of this IgG transfer across the placenta. They often, in fact, recommend that small babies only spend time around people who've had a pertussis vaccine. It's called cocooning. Weirdly enough, they tend to give older people pertussis vaccines too, in case they're around their grandchildren. At least that's why my mom and I had to fight that day. So this is one way where we're actually taking this knowledge and using it. And I heard this amazing talk about ways that people are thinking about designing vaccines for pregnant women specifically to change infant mortality, which I learned, like I said, I learned about for the first time this summer. One bad part of all of this is that if someone has an autoimmune disease that is antibody related, so here you can see a woman with Graves' disease, which has antibodies as the cause. Antibodies are causing her autoimmunity. She can actually pass those antibodies to her baby. And the baby will be born with these autoimmune issues. Because we're passing antibodies and we're not passing the B cells that make the antibodies, eventually those antibodies are going to degrade. They have a half-life. And the baby's going to be fine. It's just a short period of time at the beginning where those antibodies are present, where the protein has not yet degraded. Of course, now we know ways that you can prevent some of this from going on. So there are some things that we can do to help out here. But this is a potential issue in some autoimmune diseases. So I have 1 minute left, and that is exactly the time I need to tell you this other important fact. Okay, part one of the important fact. Wednesday and Friday, we are talking about antibody diversity. and generation of antibody diversity. It is a topic that many of you will find challenging, and many of you will be like, why did she start talking a different language today? Please, 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 do the reading before. I do not expect that you will understand anything in the reading, but I want you to have heard the words once before I say them. So please, if you have not done it any other time, this is the time I really want you to do it. And I just want to tell you what the problem is right now, and this is what we're going to spend basically the next 2 days solving. Vanessa kind of mentioned, oh my gosh, we've got all these different antibodies. I'm thinking about all the different antibodies that are out there. And if we think back to this experiment from Landsteiner, and he was showing how many things antibodies could bind, immunologists started doing some math. The math has changed over the years, but this is the same general idea. Is they find things like we can make 10 to the 16th different antibodies. As we have seen, antibodies are proteins. Where do proteins come from? Huh? DNA. They're encoded by genes, right? So in order to have 10 to the 16th different antibody proteins, you would be like, okay, so you need 10 to the 16th genes that encode that many proteins. Well, the human genome has been sequenced. That's how many genes you have. And so what you will notice is there's a really big problem here in terms of how we get that many antibodies from that many genes. And as much as I love immunology, even I will admit that you can't have all those genes be antibodies. Some of them need to be something else. And so this problem of how we get this dramatic diversity of antibodies off of such a small number of genes is going to be what we do for the rest of this week. Some of the things that you didn't like about class switch and affinity maturation today will get answered in the process of that. But be ready for this topic. And I will see you guys on Wednesday.